This is the continuity of the plasma membrane. Nature of the plasma membrane. It is mosaic. First, this is the first nature of plasma membrane, that it is has mixture of proteins and phospholipids due to presence of instructing protein. The benefit of this nature is that it is there is now method of transport across it, and the second nature is fluid nature. Phospholipid, the membrane is quasi fluid in nature, means that it within its given shape there will be movement of lipids. The lipids may carry along with them attached proteins. Lipid can show lateral movement or flip-flop movement. Lateral movement means intrinsic protein travels laterally from one side to other, or it can show flip-flop movement, that is rotation on the axis, just complete rotation. It is very rare movement. Signer said that membrane is a sea of lipids with protein icebergs. So fluid part membrane or fluidity of membrane is because of phospholipids. Membranes are asymmetric. Asymmetry in membrane is with respect to different types of phospholipids, sugar, protein in a membrane. Thickness of membrane can vary from 75 angstrom to 105 angstrom. Due to fluidity, thickness keeps changing from time to time. Fluidity, so no thickness is definite. It keeps changing due to fluidity. Mark the correct statement regarding membrane with respect to amphipathic nature of phospholipids. Membranes of fluidity. Membrane is selective permeable. Membrane is asymmetrical mosaic. Membranes have bilayer of lipids as a backbone. Amphipathic nature was the nature that allowed the bilayers to separate because they had different polarity, so they would form two layers, that is bilayers of lipid, and this due to this nature they are able to form this. Hello. Not because of the actually nature of membrane. These three are due to membrane's nature, but this is not due to membrane's nature. It is due to amphipathic nature of phospholipids. Functions of membrane are following: identification of solutes or substances in surrounding the membrane, done by glycoprotein and glycolipids, that is glyco group part. The second function is for the recognition of similar types of cell by glyco group part. Example, the ABO blood groups in our blood, they have the antibodies which will help in their recognition by other blood cells. For example, if blood group A is talked about, then antigen on RBC membrane will be A. Similarly, for B it will be B, but for O it won't be anything, and for AB it would both be A and both A and B. The antibody that A will produce would be anti-B, that is, it would produce antibodies for B. Similarly, B would produce antibody for A. O would produce antibody A as well as antibody B, and AB will produce no antigens. So it can receive therefore blood from only and only A and O, because O produces no antigens, okay, and B will produce only get only from O and B and O. O will only get from O because it has definitely, it cannot receive any blood from these two because they cannot be received by it, and AB can receive blood from anything because it has no antibodies, it has no antigens, and it has no antibodies, so it won't restrict anyone, and it can have all the now transport. This is the third function. It also has transportation in two ways, that is one water and then other is particles, solutes and particles. Water, that is simply by osmosis, it is through tunnel protein. Osmosis is the movement of, it is not through the carrier protein, it is through tunnel protein, that is the big ones. Carrier one, the small one. Osmosis is the movement of water from a region of its purity to impurity, or from dilute to concentrated, or to hypotonic to solution to hypertonic through a selectively permeable membrane. One thing very important: selectively permeable membrane transports water as well as minerals, but semi-permeable membrane transport only water. Example, cellophane and parchment. Semi-permeable water, selective permeable everything. Now, if A and B are separated by semi-permeable, that is only water, such as cellophane, then the movement would be from A to B. Oh, okay, A and B. Here has 2% NS here and 9% here. More water here, less water here. So definitely water will move from this, that is because it is high concentration here, okay, 98, and here it is 81, so it will move from A to B. But the NaCl won't move because it is only semi permeable, and semi permeable transports only water, so it will be A. But if it is separated by selectively permeable, then it would also the okay, water from A to B, very easy. Also NaCl from B to A, because it is selectively permeable, so it will also allow minerals and water. Okay. Movements of solutes is passive or active. Passive is a long concentration carried and no net ATP is used. No net ATP is used. Can be of two methods: simple either by simple diffusion or facilitated diffusion. Simple diffusion is extremely slow, very slow. Okay, and the facilitated diffusion can be a long concentration gradient. Okay, okay, and ions are transported here with the help of passive carriers called permease. Thing to remember: active transport uses ATP and against concentration gradient, and these are active carriers done by active carriers. This is done by both are simple passive are either simple diffusion, facilitated diffusion. Simple diffusion is very slow. Facilitated diffusion also also very not ATP requiring, but it is along the concentration gradient, so it is somehow faster. And with the help of passive carriers called permease. Bulk transport large amount of substances using membrane, done with vesicle method. Bulk transport is due to fluidity of membrane, due to which membrane can fold, break, join together, stretch and contract. Bulk transport is of two types: endocytosis or exocytosis, that is EPhagy. Endocytosis is two types: phagocytosis or pinocytosis. Pinocytosis is cell drinking, phagocytosis cell engulfing. Here phagosomes are found, here pinosome are formed, that is food vacuole in other words. Ephagy or exocytosis is cell vomiting or release of waste. Now, fifth function of plasma membrane is cell division. In cell division, cells resist and membrane folds towards inside. When they meet at center, it breaks into two daughter cell form. Okay, like this diagram. The membranes usually have saturated lipids, and as the temperature decreases, the membrane tends to add unsaturated lipids to increase their fluidity. This means that saturated lipids are not fluid, unsaturated lipids are fluid. So when temperature decreases, to increase their, since temperature decreases, so fluidity would decrease, but to maintain their fluidity they need to have unsaturated lipids instead of saturated